Okay, well, good morning or afternoon or night or whatever, everybody. Welcome to our fall round of the VMOL Seminar. It's my very great pleasure to have Robin, Ansgar, Corinna, and Steffen today here, who are all from the mzmine team. And we're going to kick off the mzmine season. So for the rest of the year, for consecutive seminars. They will walk us through the use of mzmine for feature finding, not only for LCMS data, but also for some other data structures. And yeah, today Robin will kick off with a non-targeted LC-MS-MS. So just to give you a very brief introduction, I've been knowing Robin since a long time, and we're good friends. Robin studied food chemistry at the University of Münster, where he then also did his PhD in mass spectrometry and drifted more towards computational mass spectrometry. He then did a postdoc in the Dorestein lab in San Diego for a little bit, before then joining the group of Tomasz Pluskal in Prague, and now is on its way to the mzio business side of mzmine. And yeah, as I mentioned, we know each other since quite a bit from the German Mass Spectrometry Society annual meeting. And we've been doing a lot of workshops together, so it's really a great pleasure to have you here and learn about the latest version of mzmine. So floor is yours, Robin.
Thanks, Danny, and thank you also to VMOL in general. VMOL is a great network that is really hosting those webinar series, but also enable researchers to collaborate across the whole globe. And so check out the videos and everything that's already available, and also sign up for VMOL so that you can follow those webinars, but also get involved in this network. And already introduced, we have a series for mzmine. There's many different topics that we want to cover, and I am going to introduce first mzmine, kind of the basics, kind of leading you how to start with the software. And I'm going to do this with non-target LCMS analysis, feature detection, and then also integrating statistical analysis, molecular networking, compound annotation, everything in mzmine. Then Ansgar is going to take over in 2 weeks. So you're going to have those seminars every 2 weeks with multimodal data processing, so mass spectrometry, but then with LCMS, GC, electron impact mass spectrometry, multi-imaging, and also other compound classes like lipid annotations. So kind of broadening the technological scope. Then Steffen is going to take over, and Steffen is the main developer who introduced ion mobility mass spectrometry in mzmine. And he's going to mainly focus on the ion mobility spectrometry processing, but then also for everyone who not yet is processing this kind of data, is also going to cover some other compound classes and maybe workflows that you might be interested in. So it's definitely interesting to tune in to see some more on different, another separation dimension. And Corinna, also in the panel here, is going to talk about automatic spectral library generation. So if you want to acquire your compounds in your lab, want to share them with other people, or just introduce your own spectral library in-house, we have tools in mzmine to process and generate MS2, MSN libraries, and anything else. And so now, from the perspective, the issue that mzmine is trying to solve is, of course, focus on the mass spectrometry. So in mass spectrometry, we often hyphenate different instruments. So we're using, for example, LC, so chromatography, ion mobility spectrometry, maybe imaging, mass spectrometry imaging here. And honestly, in the end, it's all the same sort of detector. So we're detecting the mass to charge ratio, and mzmine is using this and allowing you to process any kind of mass spec data then in the same tool. So we try to give you the same experience for, let's say, GC, MS, and LCMS, or imaging. And this comes all together by using different sampling technologies, mass spectrometers, and then also workflows, and really enabling all this data processing in just one software tool. And it is vendor data agnostic, so you can import any kind of mass spec data, either in MZML, open format, or also many supported vendor formats. And I think overall, it's always kind of a hard business that we have. We try to, on the one side, make it very user-friendly, but also make it very configurable. And then also high throughput. So there's multiple things that are kind of hard as like a technological question for us, but we're trying to basically balance all three so that you can optimize your batch in the graphic user interface. You can then go on and run it either in the graphic user interface and review the results, or you can also run it in a command line interface, so without the graphic user interface, and then just run it on a server with bigger data sets. And it all stems from like a very long history in academia. So in 2006 already, there was the first publication by the group of Matej Orisic. And then later, Tomasz Pluskal took over the project. And I mean, he comes from computer engineering. And so he really took over the project and modularized it, modernized it, and put it on a good track. And I think overall, we've been lucky with mzmine that Tomasz has been staying in the project for a long time. And now also I've been working in the project and have been leading it in the last 6-7 years. And also together with Steffen, with Ansgar, we had so many main developers basically being very stable on the project. And with this main team, we've been really driving a lot of the innovation, but we could also build on a global network of other software developers and also researchers who use the software and then also give us feedback and also develop ideas together. And so we already have now these 3 publications all together. We have just an estimate, like 11,000, maybe 15,000 active users is quite hard to estimate it really. And we have this global community with ideas and contributions. And from here, we take those ideas, we refine them, we optimize, validate, and test them. And then finally, it comes into mzmine and we ship it to our users and customers. And with mzmine, we now also have the mzmine Foundation on the one side and also the mzio company. And I think this is really like forming the company was really also an idea to keep the team together, to stabilize the project. I think we were lucky that for 20 years we've been like keeping on like developing the software, but now we actually have a company that also kind of fixes this for the long term. So you as either the academic user doing your nonprofit research, or also enterprise users, enterprise customers can really build that the software development really speeds up and that the whole product and the whole software is just more stable. And from here, we can jump into the data processing. So today we're going to do a live demo. I am just introducing all the concepts that we're going to use. Then I'm going to go through the software, and later we can have some questions for it. So first in mzmine, of course, we need to import mass spectrometry data. Mass spectrometry data either from raw formats, from vendor-specific formats, or if you already converted your files, you can also import them from mzml. Then we typically do centroiding. So if you have profile mass spectra, they're going to be centroided. So we do some peak picking on all the spectra. We also, of course, if they are already centroided, we can just do thresholding. So we can denoise the spectra and then try to find the same mass to charge ratios across the whole chromatograms. And by doing so, we build extracted ion chromatograms that are then separated, resolved into separate features. And a feature is quite an important concept here. In chromatography, a feature is going to be an ion with an mz, retention time, maybe ion mobility, and other properties. And we use those properties to then align them across all samples so that we have a matrix of the intensity of each feature across all samples. And because mzmine is modular, we can, of course, also just plug in other modules that expand into the ion mobility dimension. We can apply ion identity networking, for example, where we look at the retention time and shape of the features. And if we have let's say M plus sodium, M plus H of the same compound, they typically have the same retention time, same feature shape, and we can then group them and annotate them as adducts. Later, we're going to do molecular networking, and this can be done either directly in mzmine and also statistical analysis that we can do directly in mzmine, or of course, we can export all data and then run on GMPS, also GMPS2, and Sirius, and of course, the feature-based molecular networking statistics pipeline. Overall, if you know mzmine and if you use it in the past, maybe you know, okay, it can look much more complex than what I showed now, but this is basically just optional. There's many different steps that you can add to the workflow, but now with the mzwizard that we introduced in mzmine, you can very easily set up those workflows. So you can very easily set up a workflow where you connect LC, IMS, MS, and then data-dependent acquisition as a workflow. And we're going to do this. And before we jump into mzmine, because today we're also going to introduce molecular networking, there's been great seminars already by VMOL on molecular networking, and I guess we can link out to those on YouTube later. But here I will just introduce the concept quite quickly. So together with Louis and Danny and Ming, we've been working on feature-based molecular networking. So in feature-based molecular networking, we use the features that we detect in mzmine, and we use them and represent them as nodes in a network. And if they have MSMS, MS2 fragmentation spectra, we can score their modified cosine similarity. So either we have the same fragments, or the fragments are shifted by the modification, and we allow these shifts to get a similarity between analogous compounds. And now, very often, you're also going to see that some metadata, some matches, some annotations are also put into the networks. We could now, for example, use different spectral libraries, either from GMPS, from MassBank of North America, or mzmine, and then do compound annotation, get those matches, and then, of course, show them in the network. And matches, again, a match is usually just done by spectral similarity. So if all the different signals match and their intensity match, the cosine similarity is going to be large enough to be annotated. And similarly, we also connect in the network with a very similar concept based on modified cosine similarity. And this is just a recap again. So if we have a methyl group on adrenaline here, for example, we have noradrenaline. Of course, the precursor MZ is shifted by 14, and some of these signals may also be shifted by 14 because methyl might be retained on the ion or might be lost on the ion. And results typically look like this. So we have molecular networks that usually cluster similar compound classes together. We might have single modifications here, like a hydroxy group on these alkaloids. We may have some single substitution patterns on D and tripeptides. Maybe if someone's working with polyphenolic glucosides, you can already imagine all the different types of polyphenols, and then also how you can attach glucosides onto them. And you're going to see that, of course, this variety is captured often with molecular networking. And we can also integrate other layers of network connectivity. So this was solely based on MS2 fragmentation spectra, but people also typically look into the MS1 data. And this is what we also do in mzmine. So we look into the MS1 data, we try to find features with the same shape, same retention time, and then particular known mass differences between M plus H and M plus sodium. And if we find those, this is a real data set together with Annika Jagels. So if we then find those different ions of the same molecule, we can map it into the network, and we can then see that in a circle in red edges, we have MS1 ion identity networks, all different neutral losses and adducts of the same molecule. And then in blue, we have connected different ion species of these two compounds, and there's just a hydroxy group difference between them. And in your data set then, you might see that very many different compounds are then connected. And again, in a circle, we have all the redundancy that otherwise is kind of scattered around the network. So here, for example, sodiated adducts being connected, but being not connected to the M+H and water losses here. Yeah, so inner density networking might help you then to dereplicate your molecular networks and then also bring things together that otherwise do not cluster. So before we now jump into mzmine, this is roughly what we're going to do. So we're going to process a data set of microbial cultures. We're going to use interactive molecular networking in mzmine, do compound annotation, and then also use the statistics dashboard to connect everything. And with this, I will switch over to mzmine, and I guess you can see it. So in mzmine, if you open it, you're going to see now for myself, the latest stable release was 4.3. And I'm actually going to go to the website. Okay. Just going to type it in. So you can always go to the latest release on GitHub, and you're going to see that the latest release currently says 4.3. Of course, we're developing the software further, and I would always recommend being on the latest version because there's a lot of enhancements and a lot of new features that we're also going to introduce today. And because we have a new version coming soon, I will actually use the development release. And if you go back to releases, you're going to see that there is the latest development release. This release we don't retain. So every time we make a change, there's going to be a new release, and this is not stable. So it should be stable, but it's not recommended to use it for your analysis. It's rather like a feature release. So if you want to try out the latest features, or if you want to collaborate with us and work on a specific feature, this is most likely the release that you're going to download for the latest and greatest features. So we're going to use this today, but usually I would just use the latest release. And so in mzmine, when you start it, there's the welcome screen. You can see that there is the settings. So I can click on it and it's going to open the preferences. And honestly, many people just don't know, but there's many things you can set. You can set the formats of numbers. You can set visuals, for example, the color scale that is used. Typically, we start out with a color scale that's recommended also for color weakness and color blindness. And then, of course, there's other parameters, for example, for the data processing. For example, we also integrate with the MS Convert software, so you can also highlight and choose some settings here for the data conversion. Then we have the documentation. Documentation is very important because it's open source also. You can go there, you can learn about mzmine, and of course, you can also contribute. So if you see that something is missing, or if you feel like something could be described better, you can go to the documentation, you can go to the GitHub, you fork it, you make a little change, and it's very easy. It's all markdown, so you can very easily add information here. Then, of course, we have YouTube, where we also upload videos and where we also link out to new videos by the community. And we have the mzmine website. And on the mzio website, you're also going to find additional information for mzmine and also for the services offered by mzio. And one more thing that I want to highlight is mzmine is, of course, also open source. And there is also an introduction to development in IntelliJ. You can also use another development IDE, but you can really start developing your own feature in mzmine, and we can help you establish and realize your ideas in the software. And with this, we can now start and log in to our account. So if you don't have an account yet, it's quite easy to make one. So you can go on sign in, and then it's going to open the interface of mzio. I can, of course, I already have my account. I could now just log in. Or if I don't have an account, I can just go on register, and we only ask for very few fields to fill in. And if you provide an academic email address, so if you are an academic nonprofit user and you provide your university address, for example, you should get a 1 year academic user. And after this 1 year, you only have to validate your email again. So you just have to validate with us that you're still an academic user. And of course, for every one who's using mzmine for commercial use, you can log in here, you can sign up, and you're going to get a trial version for 1 month. And then afterwards, you could reach out to us and we can figure out how to go further. Now we can see I logged in, I have my account checked in, and we can start processing data. For this, I need to close the presentation first. Okay. So for this, I can just drag and drop this folder over here. And the easiest way of importing data to mzmine is really just selecting some samples and then just drag and drop into mzmine. You see there's a data import, and now we imported mzml files. The same is true for any kind of other formats. If I would double click on any of those files, it opens the raw data overview. And I think the raw data overview is very important, like to get the first idea of your data. Maybe you're just starting a new lab, maybe you're starting in a company, maybe you acquired your own data, but at least for me, it was always like the first thing, drag the files in here, open the raw data overview, and then maybe see at the spectral quality. So we can zoom in, we can hold control and zoom into the spectrum here on the right. We can auto scale by double clicking, and we see that it's actually already centroided during data conversion. Yeah, so the spectra are already centroided. We have chromatograms here on the side, like an 8-minute gradient. We can look at the shapes of the peaks. We can also, if we click on this check mark up here, we can also go through the different samples. And if we do this, we can also visualize multiple samples at the same time. Yeah, so we can select multiple samples. It is going to pull out all the different base peak chromatograms. This is the lines that you see. And if I click anywhere, it is also extracting the base peak as a shape. And if I want to extract another signal, I could also zoom in and, for example, look if the isotope, zoomed in too much, if the isotope is also at the same retention time. And of course, by just clicking on the isotope, I also extract the extracted ion chromatogram of this isotope. We can already see there's not much of a retention time shift. We have plenty of data points on each peak. Despite using data-dependent acquisition here, those peaks are actually quite nicely resembled. And now one more view that we can use to look at data is the data summary. We can right-click and open raw data summary here. We can use raw data at this point and OK. And this will open the MSMS scatter plot. And the scatter plot is really first like a way to see where did my instrument pick precursors and then pick the MS2 spectra. So we have the precursor MZ on the y-axis, we have the retention time on the x, and we can then see that we, for example, in the earlier range, we can see it's more distributed. And then in the later range, in this data set at least, there's a couple of different MZs that are picked too often. We can really see that there was dynamic exclusion applied because there's a distance between those MS2 spectra being picked. So there's a like 2 second, 3 second gap maybe between those acquisitions. But then those honestly, in the next run, if I acquire the data again, I would then maybe exclude these masses and just make a static exclusion list. And this is like a very easy way you can already interrogate your data. And we can also look at, of course, the other samples. This was only a QC. We can also look at the here, the 2 different strains that we have. We can look at the media blanks, and we can also see in the media blanks, it's kind of similar. And from here, we can go over to the wizard. And the wizard is really like a very easy, fast way to set up your workflows. So we can either from the welcome screen, click on this icon, or we can just go on wizard, open mzwizard. And the mzwizard, the idea is that we want to resemble something that you find in your lab and that you do in your lab. You have your sample introduction system like a UHPLC or maybe GCAE. Then you have different mass spectrometers, and you also have different workflows. And because we did UHPLC, we can select UHPLC. We could, for example, also chime in and add TIMS or traveling wave or drift tube IMS and QTOF, and then select data dependent or data independent acquisition here. So you really put together the instrument in mzmine like you do in your lab. I already prepared one preset, and this is like the most common way how I use the wizard. I set up my workflow once, I share it with my collaborators, I share it with my colleagues, and then I can just load these presets, and then everything is already configured. And in here, we have all the samples. We have an Orbitrap. So it was not a QTOF, it was an Orbitrap that we used. And we have metadata. And if you want to set this up yourself, you would basically go and say, select files, and go to the folder and select all the mzml files that you want to import. Or you could also say, I want to import all the mzml files, select a folder, and then it's also going to select all the files in this folder. For this data set, because two samples are actually outliers, it's actually good to use the select files. And I know that the QC1 and QC2 was measured first, and they actually have retention time shifts that we want to exclude. Then we have the metadata file. And the metadata file is really just a very simple text file, a TSV. And I can just open it here. So it's a very simple TSV file where we have the file name, species, and condition. And you can import it as CSV or TSV, and it's going to work as metadata. If you don't have metadata, it's very optional. You can just deselect this option. Then we have UHPLC, and it's going to capture all the different chromatography parameters. I'm not going to introduce all these parameters to all details. There's going to be Ansgar and Steffen and also Corinna also going to repeating some of this. So I'm going to focus on the most important ones now for our analysis. And I think one is, for example, the cropping the retention time range. If you still have a washout at the end of your chromatogram, or if you're at the beginning, you don't spray into the mass spectrometer, but you divert into the waste, it's definitely good to, for example, remove something in the beginning and something in the end. Then, of course, the minimum number of consecutive scans is the minimum number of data points that we want to see on our features to be actually detected as peaks. Then we have the approximate feature full width half maximum, which is, of course, the full width at half maximum of our peaks. You can either extract all these parameters, let's say, from the raw data overview by just looking, for example, for the chromatographic range here. You can also go and zoom in, for example, on one of those peaks and then very roughly estimate the full width half maximum, which is here 0.04. Or you can set it and then you can run through. And once you have the results in a feature list, it's very easy to actually get the results of a feature full width half maximum for all the features in your samples. Then there's different RT tolerance, retention time tolerances, either intra-sample, so within the sample, searching for isotopes and adducts of the same compound, and also across samples to then align the different, like the same feature across multiple samples. We have something similar when we go to Orbitrap, where we also have the sample-to-sample MZ tolerance and intra-sample MZ tolerance. It's again for alignment and also finding different ion species of the same compound. And then we also have the scan-to-scan MZ tolerance. Scan-to-scan MZ tolerance is very much like we want to find the same MZ across from the first scan to the last scan in your one sample. And of course, the mass spectrometer usually gives you that it's maybe like better than 5 ppm, better than 3 ppm, but it's usually better to go a bit wider here because sometimes just one scan might stray away from the center a little bit. And most important in this tab is the noise levels. And I think for most use cases, we can just use the factor of the lowest mass detector, which is a factor. So you don't have to optimize much. If you have a small data set, you can reduce it. If you have a larger data set and you're really struggling to process, you can increase it a little bit and it's not going to impact your data analysis that much, but it's going to speed it up drastically. Then there's also the minimum feature height. And the minimum feature height is really dependent on your instrument and on your analysis. So this is, you can kind of understand it in a way that in one sample, your feature needs to be above this noise level or this minimum height. And if this is reached, we're going to later in all other samples use gap filling to then fill in the missing features. And now to get it's actually processing, we only have 3 tabs to go. There's the filters, which is of course something like if you have triplicates, you might want to at least say that you want to find a feature in 2 samples. We actually have multiple samples per group, so I can go a bit higher. If you're scaling your analysis a lot, you could also say that 10% of your samples should contain the features. And if you're still optimizing your workflow, we typically recommend that you use only a few samples. So maybe from your 100 samples, you only take 20, maybe 10, and you set this one to keep, and it will keep all the intermediate results. So from all the different steps, you're going to have intermediate results, and you can optimize every step one by one. But if you process multiple and larger data sets, you should definitely set this to remove. And then in annotation, there's different options, and especially local compound database search is a very simple one where we have maybe MZs and known retention times, known CCS values, which you can input in a CSV or TSV database. And this is going to be introduced by Corinna in the last seminar about libraries. Lipids is also going to be introduced by Ansgar in the next session. And we're going to mostly focus now on spectral library matching. You can see that I already selected a couple of spectral libraries. There's also in the next version, there's going to be a download button where you can, for example, say we want from msnlib, the library that was built by Corinna. You can say, I want to download msnlib positive. And because I already did that, it's going to tell me to either re-download or use the existing files, and it's going to input them into this field. And you can load libraries from different vendors, for example, GmbH2, then MassBank North America, MassBank, and other formats. And now the last thing is that we set an export path. So we want to export results files. We want to export, for example, files to then be able to downstream use Sirius, molecular networking in GMPS, and other tools. And yeah, so by setting this path, you will basically generate multiple different files. And you can already see this workflow. So this is the workflow tab. Also says that we're going to apply spectral networking in mzmine. Finally, we can first of all save the preset. So I can just say save preset. You can give it a name. You can select all the different parts that you want to save to this preset and then click OK. And then it's going to appear in this list. And you can really think about this in a way that you're going to save different presets for the sample introduction, different ones for your different mass spectrometers. And then later, when you load, let's say, timsTOF positive, Orbitrap negative, you're going to load this preset and it's going to override just the parts that you saved. So now we can create the batch. And you see that now we actually have multiple different steps in the correct order. So this is a standardized DDA workflow by our team. And I can just click on OK to run it. If I go on the task, you can see that it's importing new files. It's importing some spectral libraries. And I will open the batch mode just once more. So you're going to see that there's now multiple steps that we apply in a sequence. And as a developer, it's quite nice because as long as we have multiple files, we can very easily parallelize and run this step in parallel on each sample. Then as soon as we align the sample, so as soon as we have one aligned list for all samples, we have to parallelize differently, but you see we're already here at the spectral library matching step, where we're now matching 5,000 features against like 160,000 MS2 spectra. Yeah, and this is all done on my small laptop. I hope that my like microphone and everything works fine in the meantime, because of course I set mzmine to use all. I could have limited the number of CPUs, but yeah, I set it to use everything. And it's already done. And maybe at this point, first we can look at the results, and then we can also look into some of the like power options that I just said. So the result is going to be that we have aligned feature lists, and the one with the longest name here is the latest one. Typically, we generate a name for the aligned feature list, and then we add more and more steps to it. And this also adds and makes the name longer. By double-clicking on the feature list, it's going to open the feature table. The feature table is then really like the one place where you see the ID. So if you export anything from mzmine, you're going to have a feature ID, a row ID, which you can later use to like merge results from different tools. We also have the mzio retention time across all samples. We have area, height, and I can also use the other columns here to then search for, let's say, spectral library matches that we have. And here you can see that the different colors now correspond to our samples. So we have gray is the media blanks, and in magenta we have QCs. So you can see that many of them actually cluster together, and we have different annotations done by spectral library matching. So on the left side, we have all the row types. On the right side, we have feature types. So we have types specific to each sample. So for each sample, currently we only see the height, but you can also go on the plus, and in this menu here, you can add any type of data type that we also have in mzmine. So for example, let's say the full width at half maximum, I could now add to each sample. So I can see that in each sample, we have like very similar or not so similar full width half maximum. And this I could now use for optimization of my processing. We also see the shapes here and some additional information. So you can really go into details here and kind of find out more. One thing that we're going to do here is now we have multiple samples, and we can already see that there's different sample groups here. And in the new version of mzmine, you can also recolor these. So we can go on right click, color by metadata. And if we do so, I can type in the metadata column that we imported from TSV and click on OK. And now you see that each of these groups that were specified in the project sample metadata, so we have the condition column here, each of these groups have now their own color. And this is going to help us during statistical analysis. How do we get there? We can use the quick access. So we can go select the feature list, go on the quick access, which is Control F or double shift, and then we can open the statistics dashboard. So again, we just search for statistics dashboard, and it's going to open up like this. You can see that in here, we have the PCA, we have a scores plot, we have the loadings plot, we have some box plots. And to also apply the same coloring that we did for the samples, we can just go down here and add condition in the coloring text field. Just by default, we don't show you blanks and QCs in the PCA, but I think it's actually quite nice to, at first, look at the QCs and blanks so we can just add them into this view, and we can very easily also remove them. For example, these blanks are actually media blanks, so there's a lot of different compounds in them, and they also change drastically because of the incubation and the growth of the microbes. So removing those, we can now see that the three different groups actually cluster together, so QCs and the two different strains. And we have features also corresponding to the change in our data set. So by just clicking on any of those features, we can see that some of them are annotated by spectral library match here in orange. We can see the changes here also in the box plot. And we can also directly see the annotation down here in the table. And the nice thing is really that you can change, of course, the domain PCs and range PCs and so on. You can change also over to volcano plot. So going from multivariate space to a univariate tests, we can select the condition column. We have M1 and our other strain. And now we can directly link the PCA results with the volcano plot. So if I select anything here, it's going to select the node, and it is going to select it also in the PCA. So while it is upregulated here in the PA14 group, it is also found in the same corresponding region in the PCA, which kind of makes sense, but it's a nice way of like interactively linking the different charts. And one more thing that we can also link is molecular networking. So we already processed the molecular network. So I can, for example, select any of those compounds here, and then I can go on right click and show and say feature, overview, and ion identity molecular networks. And from here, you're going to see the interactive molecular network networking visualizer. We have a spectral library match. So here, N-acetyl methionine. Honestly, first of all, thanks to everyone who make their data publicly available so that we can actually find this. But then also we have now different compounds being connected here by molecular networking. So on the left side, we have the blue edges corresponding to the modified cosine similarity of spectra, MS2 fragmentation spectra. And we also have orange edges again, connecting the different ion adducts of the same molecule. And here we can see that in this data set, we actually have methionine and also N-acetyl methionine. And of course, they are structurally very similar. But many people would now want to know why there's actually a connection between these two. And the easiest way is by selecting both. I hold Shift and select both, and then go over to the Similarity tab. And in the Similarity tab, you're going to see that there is a mirror chart that shows the precursor MZ here in a dashed line, and also shows that some signals are direct matches. So the blue signals are direct matches of the same fragment, and the orange signals are matches with the same neutral loss. So we can switch over to the neutral loss mirror, and we see that all the signals that were orange in here are now blue because now in the neutral loss, they are directly matched. And this is done by, of course, just subtracting from the precursor MZ, subtracting the fragment MZ, and then you get here, for example, 46 and 48, and you could calculate the neutral loss structure formulas. Also interesting, you can actually look at the table, and in this table, you're going to see the contribution of each individual signal pair. So you can see that in this spectrum, in these two, the intensities are actually quite well distributed, and the contribution of the maximum intense pair is actually only 0.26. Sometimes you might get a cosine similarity of 0.99, so like a perfect match. And this perfect match is really just made up by 1 in 10 signal pair being matched, and then just some spurious other signals also being matched. And in this visualizer here, the great thing is that, first of all, you can just navigate quite easily. You can change the annotation here. For example, I already said that we have multiple different adducts here. So you can see that here, for example, we have the M plus H and M plus sodium of the same molecule. We also have a couple of other things being annotated as adducts here. And if we want to see the neighborhood, for example, of this one node, I can control click on this node, and you're going to see that it's going to extract the neighborhood. And it looks very busy here. I can actually also reduce and only see the nearest neighbor. It's going to make it a little bit easier. So here we have an M plus H also being connected to a sodiated species. We have this blue node, which is actually the neutral molecule. So we try to basically visualize like a neutral molecule instead of the M plus H and M plus sodium. So if you want to, you could add a filter and then collapse away the M plus H and M plus sodium. But then we still retain the edges that these two had, the MS2 similarity edges, we still retain them to everything else being connected. And so we can go and visualize different properties here. And again, it is always just a neighborhood. So the whole complexity of your data set is usually massive. So there's usually so many relationships of compounds that are, or ions that are similar, that it gets busy quite easily. And I can control click always and just go through this network and basically focus on the next node. One thing that we can also do is we can search for annotations. So I can search, for example, maybe there are other nodes also being annotated with methionine. So I can type in something and then say, okay, focus on 2 nodes. So there's 2 nodes that actually match this name as a substring in its name. And of course, because there's a lot of different amino acids in here, if I focus on any kind of amino acid, you're going to see that there's like an explosion of the similarities, an explosion of all the different compounds being connected here. And one nice way that it's very hard to visualize now at this moment, but I can shift over the network visualizer onto, let's say, my second screen, which I cannot do right now. And I can use here, for example, the volcano plot. And if I select any node in the volcano plot, it is going to pull out the neighborhood of this feature that is selected also here in the table. It's also going to pull it out into the network and show the neighborhood. Yeah, so it's a very easy way to see, okay, what kind of compounds are significantly deregulated in my data set, and what is the chemical neighborhood of this compound. So from here, one more thing that we can look at. So statistic dashboard is quite nice and easy way to integrate these two different visualizations. But then we can also do molecular networking with different modalities. So if we go for the quick access, again, Control F, like search, molecular networking, we can start applying molecular networking also with other algorithms. So first we applied the modified cosine similarity. You can see that there was multiple different settings that we could change, like the minimum cosine similarity, minimum number of matched signals, and we can use the MS2 deep score model that we integrated together with Neek. We can now use directly in mzmine. And how can you do this? You just have to click on this download button. It is going to tell me again that I already downloaded the model. It's going to be downloaded from Zenodo. we can set up some parameters, we select a feature list, and we can press OK to then process. And I actually have to be quick. It's loading the model here, and it is already done. So it takes like a second for 5,000 features to run the embedding model of MS2 Deep Score. It's a PyTorch model. So if you're developing your models and you want to really push them out to a large community, you can work with us in mzmine, and we can host them in mzmine and make them available this way. And before we visualize this now, I first want to show you the network was already quite complex before. I pressed this full network button just a second before. It usually takes a little while to compute the network layout, depending how complex your network is. And you're going to really see that in this data set, at least, we have a lot of, let me get the annotations again. Of course, we have a lot of different, for example, peptides, small peptides, amino acids, and they are all kind of cluttered in this one network. And this is why we very much like to show you like a neighborhood. So you get one annotation, you know it's important, it's significant for your analysis, and then from there, you can go on to the next neighbors, and you can propagate these annotations to other networking nodes. And so we ran the networking. So let's open the statistics dashboard again, maybe. And from here, I can select any node. I can click on right-click, show feature overview and ion identity molecular networks. And now, of course, depending on your settings, depending on your compound classes, you're going to see that either modified cosine similarity is going to connect your compounds, or MS2 Deep Score. And in here, you can actually see that the different types of network edges, you can very easily add and remove. So I can, for example, remove the ion identity networks for now because they're based on MS1. It's not a fair comparison. And then we have the MS2 modified cosine and MS2 Deep Score. We can, again, for example, search for methionine. It's a very small molecule, so like it's, I don't know how interesting it actually is, but we could now see, if we just look at the nearest neighbors of these two, we have these compounds. Some of them are connected by MS2 Deep Score. Some of them are connected by the modified cosine similarity, and this is very much up to your settings, right? I can also increase a little bit more, and you see it is actually getting quite complex quite fast. And this was really like a success story, I would say, because with Nick, it was very nice. We integrated this model quite fast. We then made it so that you can very easily download it now from within mzmine. And they also used some data that we've been generating with Corinna. So it's a very like fruitful collaboration that we had. And from here, I think I will stop it here. The next seminars will actually have a lot of different topics, like how to optimize different settings. We didn't really go into this, but of course, now with my knowledge that I gained from my analysis, I might change some of these parameters, save my presets, and then of course, if I am happy with my batch, I would also save this batch. I would put it in my publication, upload it to Massive, for example, to Zenodo, to my GitHub, because then people can really reproduce what you did. They know what mzmine version you used, and they can also just take your data and basically reproduce your results. So with this, I will just quickly thank a couple of people. I mean, the mzmine community is much, much larger. There's like 57 contributors or 60 contributors or something now that have been working on the project over the last 20 years. Some of them are already moved on to other jobs, but many other people come back and really use mzmine as a platform to develop their ideas. We also now have received funding by the EU and also by the Bremen Aufbaubank. So it's like a fund by the Bremen state in Germany. And of course, here's some additional information. So you can find the mzmine software, you can find mzio and find out what we do and reach out to us there. Thank you.
Awesome. Well, thanks so much, Robin, for yeah, like the very nice presentation, and especially for developing this amazing tool. This looks really cool, and there's so many awesome new additions. So I'm stoked to try them out soon. Before we're gonna move on to the question and answer session, yeah, just thanks again, everybody, for stopping by, and those of you who are taking off. I hope to see you all next time.